Well, good afternoon, everybody. I'm the only one left between you and then the buffet. One of the big advantages of being the last one is that, particularly in the last presentations, already everybody explained how the immune system works. And I really have a big thank you to Per Nolan because, he saves a lot of the minutes I will have to spend in there. Immunicum is a company that in reality has started working in immuno-oncology, and Alex is there in the back of the room, in 2002, before immuno-oncology was called immuno-oncology. So it has been a long process, but really very rewarding, as I will show you. As we are listed in the main market, and I will be making some forward-looking statements, of course, I need to show you this. So several of my distinguished colleagues already explained the value of immuno-oncology and what is the objective. So we want to strengthen the patient's own immune system, whereas the normal process of killing tumor cells, to really go and attack these tumor cells wherever they are in the body. Already, several of them mentioned this is probably the fastest growing, definitely the most innovative, recognized, as Per Nolan also mentioned, with the Nobel Prizes, and is expected just the sector immuno-oncology to reach about $76 billion in 2022. So, a lot of potential, a lot of innovation. We have quite a unique approach to prime or activate the patient immune system. We do it through cell therapy. We use cells, but very important, our cells are what we call off-the-shelf, and I will show you this, what is the value. We are probably the one that is mostly advanced here in terms of development. We already closed enrollment in a phase 2 study back in January, and we are starting a new study still this year in phase 1B and then phase 2. We uplisted to the NASDAQ main market in January. We're the first company of this year, and of course, some of my colleagues also achieved that. And with that, of course, comes a series of obligations and responsibilities in terms of how we communicate to the market, and the reason why we have to be a little bit more careful. As a company, we have 3 projects, 2 in the preclinical, and ilixadencel, that is our key product in the clinical, and where I will spend most of the time. We are, the study that I mentioned to you, we closed enrollment is in kidney cancer, renal cell carcinoma. We also finished last year one study in liver cancer, hepatocellular carcinoma. We closed enrollment in orphan disease, GIST, a project that we are collaborating with the Karolinska, and a new study that we are initiating, head and neck squamous cell carcinoma, non-small cell lung cancer, and gastric adenocarcinoma. And this is a study. As a company, we have a product is approved by the FDA for clinical trials since 2016, and for this new study, we got the clearance from the FDA back in July and ready to start enrolling patients. So where does Immunicum and ilixadencel fits in what we call the cancer immunity cycle? Different classes, Per Nolan already mentioned that, the checkpoint inhibitors and the PD-1s and the PD-L1s, the kinase inhibitors, some of the chemotherapy, they are on this side of the equation. This is the tumor blocking the defenses. The big problem is that if the patient's own immune system is not active, then there's a lot of really non-efficacy from these classes. So you need what is called make the tumor hot, bring these immune cells to the site of the tumor. That's what we do. I decided to use this time a very scientific illustration because, I needed to compete with my colleagues. So I normally use the analogy that the tumor is like a castle, a fortress. So creates the defenses, and the patient's own immune cells cannot penetrate. So the classes of drugs that was already mentioned, the checkpoint inhibitors, the ones that are commercialized, and kinase inhibitors, their role is to block the defenses. In this case, destroy the entry gate to the castle. but normally with a relatively small-sized army and not the strongest one. So what we do, and I will show you how, is that we bring the additional troops. So we activate the patient immune system. We increase the number and the strength of this army so that the army goes through the gate and destroys the tumor. And destroys the tumor not only at the site of the primary tumor, but also in the metastasis, because the tumor creates some other more peripheral posts that we call the metastases. So this is what we do. We bring a stronger and more activated army to destroy the tumors. Key differentiation to a lot of other cell-based therapies, as I mentioned to you, we are what we call off-the-shelf. These cells are originated from healthy donors. These are like blood donors. We extract the cells. We don't do any genetic manipulation, any expansion. We just activate them with a cocktail of cytokines and make these cells extremely pro-inflammatory. So they are going to behave, although they are not, like a vaccine when you inject. We currently manufacture in Germany, and we were distributing in this phase 2 to 28 centers in 8 European countries and the US. Very important here, from one donor, we extract about 100 doses. We treat each patient with 2 or 3 doses, depending on the tumor. So, from one donor, we treat up to 50 patients. So this is, from a perspective of the cost of manufacturing, has really no impact on the margins. Here, again, another key differentiation is that the product is frozen, sits in the hospital pharmacy. We already passed 3-year shelf life. And the product is what we call universal. It's going to be the same cells that are in this little vial, 1 milliliter, and these cells are going to be the same ones regardless of the patient X or Y and regardless of the type of tumor. So this is very important because you don't need to do any matching beforehand. The product that sits in the pharmacy is the same regardless of the type of tumor and regardless of the patient. Sits in the hospital pharmacy, and what happens when it's needed? You just put it in a bath of 37 degrees, body temperature, and then you inject into the tumor. And we only need to inject one mass. We don't need to be injecting different masses. Can be the primary tumor or the metastasis. It doesn't matter, because the mechanism of action is basically when these cells from the donor are injected into the tumor, they start producing all these inflammatory mediators, chemokines and cytokines, and what they are going to do is recruit the patient's own immune cells to the site of the injection. They are going to be activated by these chemical mediators, kill the tumor cells, release the neoantigens that is like a kind of a barcode for that specific tumor in that specific patient. The patient dendritic cells take on this barcode, go to the lymph nodes, and then train there the important cells to kill the tumor that are the cytotoxic T cells, CD8 T cells. These are that army that then spreads through the whole body and going to attack the tumor cells regardless of where they are located. Can be the primary tumor or can be the metastasis. So very important here, is a universal product, but when you inject it, it becomes a personalized product. We are already quite advanced in terms of clinical development. This was the first study that was already done, initiated in 2013. And we did it in newly diagnosed and metastatic kidney cancer patients. So the patients were injected with 2 doses, 2 weeks apart, and then if the kidney was removed, and then if they will progress, they will be treated with the standard of care. So what you see here is basically, these dark dots are the normal, those T cells, killer T cells, the normal, what you see as concentration as a reaction to the tumor. And what you see here, this is in the main tumor, the extensive infiltration of these killer T cells in the tumor. Tumor specific because this is the healthy tissue just outside, you see the normal, but very important also in the metastasis. So really these killer T cells spread through the whole body. Importantly is also in terms of the clinical efficacy. This study already finished, as I mentioned to you, long ago, but we still had in January 3 out of 11 patients still alive, already passing the 5-year survival. This study was published last year, and we reached a median overall survival of 48 months, 4 years, and the normally expected with the standard of care is 15 to 16 months. So it's not a big number of patients, but quite an extended survival, and the fact that we still have 3 patients alive past already the 5-year mark. One of those patients in this, we use this case just to show the value of the combination, because today the treatment of cancer in immuno-oncology is with the use of combinations, classes of drugs that have different approaches. As we saw, the ones that destroy the gates, the ones that then activate and destroy the tumor. This patient had a poor prognosis, survival expected 9 months. So we had several liver metastases, several lung metastases, and then he developed 4 brain metastases. And, you know, at the time, this is a patient, again, as I mentioned, from 2013. His expected survival was 9 months. He was used the combination. Sunitinib, Sutent, was the drug standard of care at that time. Well known that doesn't cross the blood-brain barrier, so doesn't destroy the brain metastases. Anyway, this patient in January was still alive after 56 months. The brain metastasis disappeared, the liver metastasis disappeared. He had 1 small lung metastasis that was removed surgically. So, you know, from 9 months expected survival, still alive after 56 months. Is one case, but is the value of showing the combination of these different mechanisms of action. The patient had so many metastases that he needed the standard of care to reduce the number of tumor cells and gave opportunity to the patient immune system to then go and destroy in the locations where the therapy doesn't arrive. After the study, we initiated a phase 2 study. This is a study that we concluded the enrollment in January, 88 patients, and this is a study that is controlled. So we have 58 patients that use our cells plus the standard of care, sunitinib, versus 30 patients with sunitinib alone. We are looking at survival, 18 months, so we are going to have the top-line results 3rd quarter next year, very, very soon. And here we will be able, it's a very important study, as also some of my colleagues mentioned, because this will give you the data that a lot of the potential partners are waiting for, controlled data showing the combination versus the drug alone. We also have a couple of publications waiting. But the main study now project at the moment is this new study, what we call now ILIAD. The landscape, the treatment patients have evolved to use checkpoint inhibitors. As several of them mentioned, these are several drugs, the Keytrudas, Optivos, you know, drugs in the market that have a relatively poor efficacy in several of the solid tumors. So this study, we got clearance from the FDA, as I mentioned, in July. Centers are being activated, and we expect the first patient to be enrolled before the end of the year. The study is going to be run in the US. And the main reason for that is because the checkpoint inhibitors are standard of care in the US. So these patients are going to be treated with a checkpoint inhibitor anyway. We just add then ilixadencel. Of course, this helps us because we don't need to pay for the checkpoint inhibitors that are quite expensive. We had several discussions with the European authorities, but also the FDA. We significantly increased the number of patients and the design of the study. So the phase 1B will have now 21 patients. This is going to give us already next year very important data in terms of the safety of the combination. I have to mention also that ilixadencel has an excellent safety. Basically, the side effect that we have is fever that we treat with paracetamol. Because the donor cells that are injected are killed in the first 2 to 3 days, and so is the patient's own cells that are then impacting. So here we also had to disclose, we asked by the NASDAQ that we are in discussions with a pharma company for them to supply us for free checkpoint inhibitor. The big value, in addition to the scientific validation, is the fact that then we can extend the study from the US to European sites. I want to spend the rest of my time on this slide because it's, of course, the big news from last week. We, of course, one of the benefits, but also the downsides of progressing and success is that you to need more investments and bigger investments. So we are very happy that the development and the milestones that we have been reaching as a company, as we promised the market, have been recognized by a series of very important institutional investors. I think if you look at the list, you recognize it's probably is the crème de la crème of the institutional investors investing in biotech in Sweden. So this is very important, and they will, in a directed issue, secure 178 million. It was important for the board and for these investors that we also, the existing shareholders, have the opportunity to participate in the same conditions. We believe that our current shareholders will see the value not only of what we deliver, but what we are going to deliver with these investments, and they will participate. But of course, it's the job of the board to make sure that we have this cash, and it's 100% secured. But here I want to mention that, different from our last capital raise, is an excellent group of guarantors. As a matter of fact, we were able to secure 100% at very, very attractive conditions. And some of them even really like the project and are going to participate in the subscription. So this also shows how they believe in the project. So in total, about 351 million SEK that are going to be used in projects, clinical, but also the development of a commercial process that are enormous in terms of the value that they will create to the company. So you see there the dates. You will continue to hear from us. We are here. We'll be presenting to the investors and the current shareholders. So as a company, we have been achieving all the milestones that we have committed to deliver to the market. We have a clear strategy and a plan implementing it. We assembled, quite a knowledgeable team that is very important in this innovative area. So excellent growth opportunity for the company. Extremely important, the results of the phase 2 study that in a way we are obviously, as we are all in discussions with big pharma, the usual suspects in this field that basically are just waiting for the clinical data. So I already overextended my time, so I want to thank you everybody for your attention.
Thank you, Carlos. You didn't overextend your welcome, that's for sure. Firstly, the fact that ilixadencel is an off-the-shelf product. What do you see as what are the key advantages of this, as opposed to the more personalized approaches?
It's really, several big advantages. One is, as you know, for instance, everybody's familiar with the CAR-Ts, although CAR-Ts are not in non-solid, the autologous approaches, is the cost and the complexity of the logistics. You need to do a biopsy, select the neoantigens that you don't know if they are going to be the right ones. So it's a lengthy and very expensive process just to manufacture. And you need to have quite a complex logistics because you need to be shipping product and samples all over the world normally from one center. So we don't have that complexity. So the cost of goods has no impact on the margins of the product. And basically, we can produce whenever we want because as we already passed the 3-year shelf life, so, then at the moment we have product in the US and then just sits there when it's needed, is readily available. And this is a big difference. So if the patient needs it, the product is there instead of having to wait for the more personalized. But when you inject it, then it becomes personalized for that type of tumor in that patient. And that's the other big advantage.
Are there any downside to that?
Not really at the moment. I think we have now close to 100 patients already treated in different types of tumors. I think normally the big concern is in terms of safety, particularly because of some of these classes of immunotherapies in terms of safety. So that is we have excellent safety. So as a matter of fact, we now with the new study, we are going to have data, 6 different types of tumors, combinations with 3 different classes of drugs. So a huge potential if we replicate the data, for instance, the results that we had in the phase 1b.
And the Iliad trial, what are the main advantages then of running that trial as 3 smaller studies in different indications, as opposed to in just one larger in single indication?
No, thanks for that question, because I think the new studies in oncology, particularly in immuno-oncology, have been moving to what is called adaptive design. So we have the phase 1b that will have the mix of the 3 types of tumors, but when we move to the phase 2, we are basically going to run these 3 indications in parallel. One of the big benefits and one of the reasons why we selected these indications is that they are very big, but we know there's a lot of data that we know that the efficacy of the checkpoint inhibitors is very modest. For instance, head and neck is less than 20% efficacy. So this means that we don't need a lot of patients in each of the indications if we have a difference, to show that we make that difference. As a matter of fact, in the design, we have a very early reading. If 1 out of the first 7 or 9 patients, depending on the type of tumor, reaches a predefined response, we can continue. Okay. But if not, then we kill that indication. So this is very efficient in terms of cost and in terms of not being treating patients where there's not going to be efficacy. So, but of course, from an investment perspective, diffuses the risk, because we basically have 3 shots at goal.
Yeah, and speaking of investments, then, like you mentioned, you're raising quite a lot of capital right now, and roughly half of that 351 million Swedish kronors come from institutional investors. Some of you call it the creme de la creme of biotech investors in Sweden. How will this affect Immunicum as a company going forward?
I think has been, I'm really very happy that, of course, the developments of the company gave them this confidence that we have been delivering on what we promised and the enormous potential for the future. But this what provides is, as we all know, the big challenges is when you need cash, maybe the cash is not available because you never know how the markets react. Yeah. So the fact that we have these strong investors, and that are ready to reinvest if there is a rationale for that, and the fact that with this capital raise we are basically funded towards the end of 2021 allows us to make these very big investments, but very big value driving investments, but also provide the stability that the company can really focus on the activities that drive value. That is, moving along with the development of not only the clinical trials, but of the product, instead of being continuously in small rounds of financing.
Chasing money.
Yeah, and we probably will be talking about this, in the Q&A session. I've been, since I joined the company 2 years ago, presenting the company also in the US and in Europe. And there's always one typical question. Who are your top shareholders? And so it's very important for any company to secure a consortium of big investors that then will facilitate and will make it easy to be accepted as an investment opportunity also outside, in this case of Sweden, from the US or from Europe. So there's definitely different benefits.
Yeah.